All right, welcome everybody to this third day of the SciLifeLab Outreach Week. My name is Magnus Lundgren. I'm a platform coordinator at the genomics platform of SciLifeLab, and I will be the moderators for today's session on evolution, molecular ecology, and environment. So we're going to very soon start off with an introduction to the topic and the services in the field provided by SciLifeLab by Olga, Vinnere, Pettersson. And then we follow a number of scientific cases, very prominent researchers and very interesting research questions being addressed. Just a couple of technical comments. As you noticed, this session is being recorded, and for those speakers who have agreed to do so, this recording will be made available on the SciLifeLab YouTube channel. There will be a possibility to ask questions through the chat or directly to the speakers after their presentation as far as the time allows, but there will also be a specific Q&A session at the end of the session where you're also welcome to ask questions. So without further ado, I welcome Olga Vinnere Pettersson, who is a biodiversity coordinator at the National Genomics Infrastructure at SciLifeLab, and also a project coordinator at the same unit. So very welcome, Olga.
So good morning, everybody, and I hope I'm heard and you can see my presentation. Magnus nod. Excellent, thank you so much. So my duty today is to present the SciLifeLab infrastructure in light of today's topic. And actually, I'm celebrating my 10 years with SciLifeLab this year. So it was really exciting development to see how all these technologies are coming together and what kind of services SciLifeLab can provide. I guess if you are here, you know what SciLifeLab is. It's a huge pan-Swedish organization, which is striving to provide Swedish researchers with world-leading technologies so that Swedish researchers can then produce world-leading research. Physically, we're located all over Sweden. However, the main operations are in Uppsala and Stockholm, but we have a distributed model where many different facilities are spread all over the major Swedish universities. We have facilities in Örebro, Umeå, Linköping, Lund, Göteborg, Umeå, and so on and so forth. SciLifeLab has three main pillars or three main ideas of organization. So first of all, we're providing the infrastructure. So this is a type of equipment that would be too costly or too complicated to maintain at different universities. So we're providing that more or less centralized. Then, of course, SciLifeLab associated fellows and researchers are doing lots of work in studying different aspects of life sciences with help of the infrastructure and the technologies. And then finally, recently, we launched a data-driven life science program with generous support from the Ballenberg Foundation, where all this huge amount of data that SciLifeLab is produced are being structurized and witnesses identified, data streaming is enhanced, and so on and so forth. If we're looking at all the SciLifeLab platforms, we have quite a few of them. Very often, as a biodiversity project coordinator, I hear that SciLifeLab is very focused on human research and biomedicine. However, that is not entirely true. Within the SciLifeLab, there are lots of platforms that can be of great use to the biodiversity researcher, to evolutionary biologists, infection biologists, and so on. As you can see here, we are trying to promote the planetary biology, which is molecular ecology and all the other types of biology aspects that can be applied for studies of ecosystems on our planet. We have also a program in systems biology, structural biology, and of course, the data-driven life science. If I start to present each platform separately, I should perhaps start with my own technology platform, which is genomics. This is the largest platform within the SciLifeLab, and also later today you'll hear lots of the users or researchers that have been users for the genomics platform, but also we're combining their research with other types of services that can be provided via SciLifeLab. So SciLifeLab Genomics platform is the third largest next generation sequencing core in Europe. And it consists of the NGI, ancient DNA, and microbial single cell genomic platform. What we can help you with is advanced user support. And then we take everything from the project design down to the sequencing and primary data analysis. All the new users are very welcome, even if you do not have any previous genomics experience. We have a wide array of specialists who could guide you through this jungle and tell you what is the best application for your particular project, what is the best sequencing technology, and also give you some hints how you can analyze the data. Also, we can connect you to other parts of SciLife infrastructure who could do the downstream analysis. Also, we can provide all the guidance in sampling, and in some cases, we can also do nucleic acid extractions. For the ancient DNA, we also provide both extractions and library construction. And for reference genome projects, we can extract high molecular weight DNA and RNA. If we're speaking in particular the first part of the genomic platform, which is the National Genomics Infrastructure, we have services to cover from a single SNP in your genome down to providing the reference genomes for whatever organism you would like to study. Recently, we started to provide a complete package for reference genome sequencing from extraction to the draft assembly. We can provide lots of different services in the field of metagenomics, which can be from amplicons from the single gene to the sophisticated barcoding, which is amplicon-based. We can also do shotgun metagenomics, both on short-read technologies like Illumina and long-read technologies like PacBio and Oxford Nanopore. Biodiversity studies are impossible without population genetics and genomics, and there we can provide you with both SNP arrays and with sequencing. In terms of transcriptomics, we have the full array of services. We can do everything from microRNAs and ending up with whole-length transcripts for new isoform discoveries and annotation, and also provide a direct RNA sequencing on Oxford Nanopore technology for discovery of RNA modifications, if you're so inclined. We can also provide you with full range of epigenetics that can be both with SNP analysis and with sequencing, and it can be done both on short and long read. And also we have quite an extensive R&D. If there is a new technology in the world, we're trying to adopt it, understand how it works, to try to figure out the pros and cons and then start offering that as a service. And at the same time, very often when a scientist comes to us and say, we have a scientific question and we do not know how to study it, or we have a good idea what kind of NGS application they would like to have, sometimes we can go all in and help researchers to develop such an application if we feel that that is something that can be beneficial to the entire community. And how to contact us. If you have any questions, that's the best if you book a meeting with us, and that you can do through the genomics portal. Meetings are non-binding and they don't cost anything. We're here, we're happy to serve you, and the sole purpose is to help you to conduct a world-leading research. The ancient DNA unit is focusing mainly on DNA from the past. So that is, if you have a herbarium material, or if you have an ancient mammoth sample, or if you have ancient burial grounds, this is something that Magnus facility or ancient DNA unit could help you with. They can try to help you with analysis of bone material and soft tissues, sediments, hair, and so on and so forth. Everything is done in clean rooms, and they can help you from the DNA extraction to library construction. And then the libraries will be passed further to the NGI, and NGI will do the sequencing. So there are lots of different types of analysis that the ancient unit can provide you with. Kinship can study microorganisms, look at the degree of preservation, try to validate the age of your specimens, and so on and so forth. And the contact for them is via SciLifeLab homepage. And our Magnus, who is the host for this part of the program, he's actually the head of unit. So you're most welcome to direct your questions to him. Another very exciting platform, which is a part of SciLifeLab Genomics, is Microbial Single Cell. And they're focusing both on the pathogenic organisms and also on the wide array of metagenomic research. The pathogens can be studied in BSL level 3 laboratories. It can be anything from infection biology and eukaryotic parasites like Giardia, for instance, or malaria, or it can be also viruses or pathogenic bacteria. But in terms of metagenomics, that's a full services of the shotgun. And the most important is also single cell sorting. So now we're approaching the time when we can sequence the reference genome of microscopic eukarya with help of microbial single cell facility. And you can find information about them again via the SciLifeLab homepage. The next very important part of the SciLifeLab platforms and SciLifeLab infrastructure is, of course, data analysis. And here you can get help from the Bioinformatics platforms. To the Q&A session, we'll have Sebastian De Lorenzo joining. So if you have any specific questions about the ENVIS, you're most welcome to contact him. But basically, when SciLifeLab Genomics is producing data, we are shipping it further to the ENVIS platform. And ENVIS platform can provide you with a huge array of different types of analysis with statistics, metagenomic analysis, assembly and annotation, gene expression studies, population genomics, and so on. They can also help to develop web services and databases. And very importantly, in light of the new DDLS program, they can help you with data management plan, metadata collection, and so on. They have 3 different support models, which are either based on the user fee, or you can also apply for long-term support, where you get bioinformatician for free up to 2 years, working closely with your group. And every week they have free online drop-in services where also genomic specialists are present. So you're most welcome to drop in, doesn't cost anything. There are lots of very knowledgeable people that can guide you through different aspects of the analysis. And they have their own website, envis.se. And there you can also apply for support from the Bioinformatics platform. It's very difficult to study biology without actually seeing what you study. And hence we have cellular and molecular imaging platform that is providing with cutting-edge microscopy and visualization tools for any part of your research. There is a huge array of different types of electron microscopies, and this facility is evenly distributed across Sweden. For instance, there are very strong facilities at Umeå University in Stockholm and so on, and you're most welcome to contact them via the SciLifeLab homepage. If you would like to study metabolites in your biological samples, here is metabolomics platform that is for you. So you can study everything from lipids and small molecules to proteins, and you can look at the interactions between different types of organisms you're working with, or look at the secondary metabolite profiles, and so on and so forth. The homepage is at Swedish Metabolomics Center, and Annika Johansson is the head of unit. So you're most welcome to contact her. And then, of course, exposomics, that is the summary of all the environmental exposures that your organisms or systems can come in contact with. And here we have a special unit dealing with that. It's a new word, it's called exposomics, but it's perhaps important for you to know about. The next facility, it's called clinical proteomics and immunology. But if you are studying poisons, or if you are studying different types of proteins that that is also something that they could help you with, despite their name of being clinical. So for that, you could contact Masoud, and he'll be more than happy to sit down and discuss a project with you. And finally, I would like to close with my last slide. I would like to highlight some of the collaborations that SciLifeLab is having, and that is Earth Biogenome Project, aiming to sequence all eukaryotic life on the planet with a focus on the conservation biology, and also its European node ERGA. If you would like to know more about that, you're most welcome to contact me. And that is it. And I'll stop sharing my screen.
Thank you very much, Olga. That was an excellent presentation overview of the SciLifeLab services and organization. So next up is our first case study, a presentation by Dahlén. He is a professor at the Swedish Museum of Natural History and also a research leader at the Center for Paleogenetics, which is a collaboration with the Swedish Museum of Natural History and Stockholm University. So will tell us about how early Pleistocene DNA can unravel the genomic history of mammoths. So very welcome, Love.
Thank you, Magnus. I'm just going to share my screen here and start the presentation. Hope you can see it now. Right, thumbs up from Magnus. All right, I'd like to start by thanking Magnus and SciLifeLab for inviting me to give this talk. And let me see here. As Magnus said, I'm going to talk to you about essentially a paper that we published last year. That has to do with a genomic history of mammoths in general. And as Magnus also already said, I'm working at the Center for Paleogenetics in Stockholm, which is this collaboration between the Swedish Museum of Natural History and Stockholm University. So I'm going to start by telling you a little bit about really old mammoths. I guess what most people think of when they think about mammoths are the species that existed during the last ice age, primarily the woolly mammoth, which you're all familiar with, but also the Colombian mammoth, which is a second species that inhabited the lower 48 states of the US, as well as down into Mexico and Costa Rica. And so if we start and have a look at what has been believed about the evolution of these species of mammoths, we have to rewind the tape all the way back about 5 million years when the very first mammoths appeared, actually in Africa. And these mammoths, the earliest members of the mammoths genus, they relatively soon left Africa and expanded into Eurasia. And if we are talking about something like 3 million years ago, we have a species which you can see to the left here called the southern mammoth, Mammuthus meridionalis. If you go to the Swedish Museum of Natural History, there's a mammoth there in the exhibition, or it's a cast of a mammoth, and that is actually a meridionalis southern mammoth. But so the idea of mammoth evolution is that these southern mammoths, they eventually evolved into another species that was called the steppe mammoth, Mammuthus trogontheri. And then the sort of classical model of mammoth evolution is that these steppe mammoths gave rise to both the woolly mammoth and the Colombian mammoth. And the general thinking is that this happened, this speciation events happened sometime between 800,000 years ago and 1.5 million years ago. And so we have for a long time been interested in trying to disentangle this evolution and study what really happened during the speciation event. But part of the problem with that is that if we want to do that using ancient DNA and study this process sort of in real time, we would have to go back and get DNA from probably beyond 1 million years ago. And that's been a bit of a challenge, given that the previous oldest DNA ever recovered was about 650,000 years old. Now, fortunately, mammoths inhabited cold regions of the world, and in particular, the speciation events involving the origin of the woolly mammoth as well as the Colombian mammoth took place up in northeastern Siberia. And in Siberia, you have thick layers of permafrost. This is a picture from the Batagaika crater in Siberia, where the permafrost is hundreds of meters deep. And DNA is exceptionally well preserved when it is preserved in cold state, for example, in permafrost. And so here's a photo of Andrei Sher, who was a famous Russian paleontologist who sadly passed away in 2008. But I had the fortune of working with Andrei in 2007 when I did a Marie Curie fellowship in London working on mammoths. And already back then, we started discussing these really old mammoth samples. And the idea was that maybe we could get DNA from them. But back then, we felt that the techniques available with PCR and Sanger sequencing weren't really up for this kind of analysis. But we picked this up again a couple of years ago, and we then had 3 specimens from Andrei that we focused our efforts on. And you can see photos of these 3 mammoth teeth here. One of these specimens was about 700,000 years old from Chukotka. The second specimen from Adycha was about 1.1 million years old. And then finally, we have the Krestovka specimen that is roughly 1.2 million years old, according to stratigraphy, to the geological dating. And so one of the PhD students in my group, Patricia Pechnerova, she set out to extract DNA from these samples and eventually sequencing them at SciLifeLab. And when we just quickly look at what the DNA looks like, you can see this figure here. This basically shows on the y-axis the number of reads mapping to the reference genome, and then you have on the x-axis read length. And the red part here are reads that we think are too short to use for anything. They are simply unidentifiable. And the black part are reads that we trust. And as you can see here, the DNA in these specimens is extremely fragmented down to an average size of between 40 and 50 base pairs. If we compare this to much more recent mammoths that are between a few thousand years old and up to 40,000 years old, so orders of magnitude younger, you can see that the DNA in these more recent specimens is much more well preserved. But nonetheless, it was possible to extract these minute amounts of DNA reads that we could confidently say are mammoth and proceed to work on them. And essentially, we were able to reconstruct genome-wide data from all these 3 specimens, ranging from something like 70 million base pairs from one of them up to a couple of gigabases for the one that we got the best data from. And you know, one of the first things you want to ask yourself when you do this kind of studies, you know, are these specimens really this old as the geological dating indicates? And so, to cut a long story short, the answer is yes. We performed, to investigate this, we performed molecular dating of the specimens, using basically relying on the molecular clock, using both mitochondrial DNA and for 2 of the specimens also nuclear DNA. And if you look on the right-hand part here in the figure, you can see the red bars are the molecular data estimates and the blue parts are the geological data estimates. And roughly speaking, they corresponded to each other. So we now could feel fairly confident that we had DNA on the order of well above 1 million years old for 2 of the specimens. And so if we then look quickly at the origin of the late Pleistocene mammoths, the woolly mammoth and the Colombian mammoth, we initially just built a simple phylogenetic tree. You can see Asian elephant on the top here, and then our various mammoths below. So at the very bottom of this tree, you have late Pleistocene mammoths, Wyoming, Scotland, Omiaco, Kanchalan, and Wrangell. And as we expected, Chukotka, which is about 700,000 years old and represents one of the earliest known woolly mammoths based on morphology, also is based basal in the phylogeny of woolly mammoths. And then we have the steppe mammoth, Adycha, which seems to be ancestral to all the woolly mammoths. But somewhat to our surprise, the Krestovka specimen was much more divergent and fell basal to even the Colombian mammoth. So that was quite interesting. And we wanted to investigate this further. And one of the things that you then can proceed to do is to use these statistics. And asking the question specifically about the Krestovka mammoth, which in the phylogeny is basal here, we can ask the question, you know, based on this phylogeny, the Krestovka specimens should be equally ancestral to all other mammoths. And we can test this using these statistics. And what we found was that was not the case. Instead, we found an excessive allele sharing between the Krestovka specimen on the one hand and the Colombian mammoth on the other hand. And so that was quite surprising, and we kept investigating this using other methods, and one of those is mixed graph modeling, which was, we got significant help to do this from Pontus Skoglund and Anders Bergström over at the Crick Institute in London. And again, to cut a long story short, what we now think we are seeing is that the Colombian mammoth is actually a species that originated through hybridization between this Krestovka type of mammoth on the one hand and woolly mammoth on the other hand. And so that's pretty cool. We think now that the genetic contribution to the Colombian mammoth is about 50% from each of these two species. And to further test this, Tom van der Walt, who's the first author on this paper, he proceeded to do a ghost ancestry analysis, which is an analysis you do without involving any of the ancient specimens, just looking at the Colombian mammoth. And you can see here that there's kind of this bimodal distribution where you have the unadmixed part that looks like woolly mammoth, and then there's this ghost component. And when Tom then pulled out those reads and put them into the phylogeny, lo and behold, the ghost regions actually group very closely to the Krestovka mammoth, thus providing somewhat independent evidence of this hybrid nature of the Colombian mammoth. So to sum that up, we do think we found evidence for a previously unknown lineage of mammoths in Siberia, represented by the Krestovka genome. It might very well represent a hitherto unknown species of mammoth. And secondly, we also find evidence for that the Colombian mammoth seems to have originated through a hybrid speciation event, maybe sometime around 500,000 years ago, through hybridization between woolly mammoths and this unknown Krestovka lineage. Finally, I wanted to tell you a little bit about another aspect of this study, was that since we had this Adycha mammoth, not the Krestovka, but the other, the Trogontheri-like mammoth, which seemed to be directly ancestral to all the woolly mammoths, and we also had a woolly mammoth that was about 700,000 years old, and a bunch of woolly mammoth genomes that were much more recent, up to 4,000 years old. We have this kind of genomic time transect spanning over 1 million years. And so one thing that we could look at, in addition to the sort of phylogenetic relationships, is adaptive evolution. And so we wanted to ask the question, when we have a time transect spanning over 1 million years, what can that tell us about the rate of adaptive evolution through time? And so we did this looking both at generally across the whole genome, non-synonymous mutations or protein coding changes in the genome. And we also looked at the number of genes, 91 genes that had previously been described by Vincent Lynch's group over in University of Chicago as representing adaptations to life in the high Arctic. And so if we look at this, obviously in the late Pleistocene woolly mammoths on the right, we expect 100% of these mutations. That is a guaranteed outcome. But when we look then at the overall non-synonymous mutations in these genomes, we find that already over 1 million years ago, 85% of these mutations were present. So you can basically draw a straight line through this, suggesting that the rate of non-synonymous changes in mammoths over 1 million years happened at a relatively constant rate. Although, of course, these are not that many samples, but based on these 3 time points. And somewhat to our surprise, we find a similar pattern for the Arctic adaptations. I would have thought that when the speciation happened, when Mammuthus trogontheri evolved into Mammuthus primigenius, the woolly mammoth, we would have seen a sort of rapid rate of adaptive evolution in terms of adapting to the Arctic, but that's not what we found. And if we just quickly look before I end at these steppe mammoths, quite little was known about Mammuthus trogontheri before this study. We knew what their skeletons looked like, but not much more than that. And so based on some of these Arctic adaptations that we see, we do think that they were already adapted in terms of hair growth and thermal sensation, fat deposition, thermoregulation, and circadian rhythms. So we could then ask a paleo artist to draw a sort of revised reconstruction of what these steppe mammoths might have looked like. given that they likely were highly adapted also to an Arctic climate. And with that, I would like to thank you for listening, and also just quickly to thank the lead authors on this paper, which is Tom van der Valk, Patricia Pettersson, David Diaz del Molino, and also our current PhD student, Marianne de Haske, who did a substantial amount of work on the Kestovka specimen. Thank you for listening.
Thank you very much, Love, for that excellent overview. I'm very happy to, on behalf of SciLifeLab, to provide sequencing for this project. We could have time for 1 or 2 questions to Love, if there's anyone who have something pressing on their mind. You can type it in the chat or just unmute yourself and ask the question. I could just go maybe for one sort of general question. So on the nature of your interaction with SciLifeLab, if you have any thoughts or comments on the usability of our services for this project?
Well, obviously, SciLifeLab has been, and in particular NGI, has been absolutely essential for this work. I mean, it's been we have a long collaboration with SciLifeLab in terms of the DNA sequencing, and you know, we have, I think, at both ends learned a lot from this interaction, and so that we were really ready to go when we started this project. So everything, all these ancient, the really old specimens were sequenced at SciLifeLab in Stockholm.
Okay, thank you very much. We have another couple of questions in the chat, so if you would like, Love, you could respond to that. So then we welcome our next speaker, Associate Professor Nathanial Street from Umeå University, the Department of Plant Physiology. So very welcome, Nathanial.
Hopefully you see the presentation and hear me. Yep, great. So thank you very much for the invitation to present today. It's great to get the opportunity to feed back and present what we managed to do with all the sequencing data that we generate using SciLife and NGI. So today I'm going to present a project where we've been using RNA sequencing to study the interaction between forest trees, so this is specifically Norway spruce today, and associated ectomycorrhizal fungal species. So we're studying Norway spruce. Norway spruce lives in the boreal forest, and the boreal forest is the largest terrestrial biome in the world. And in terms of carbon storage globally, the boreal forest is hugely important. It's the largest carbon store. We're all quite familiar with thinking of tropical tree species and how important these are globally, and rainforests and things, but the boreal forest is really hugely important for carbon storage. In the boreal forest, a characteristic is that tree growth is very limited by low nitrogen availability. And importantly, up to 70% of the stored carbon in the boreal forest is derived from tree roots or plant roots and root-associated microorganisms. And the root-associated microorganisms are a large percentage of that stored carbon. So the interaction that we've been particularly interested in is between ectomycorrhizal fungi and the roots of trees. And these are fungal species that kind of encase the roots, and they enter into a mutualistic or beneficial interaction with the tree, where the tree is supplying sugars, so it's supplying carbon, so the fungal species, and the fungi are producing and supplying, or they're making available nutrients. And importantly, in the boreal forest, this is particularly nitrogen that we've been interested in, but they're also very important for making phosphorus available and also for water availability. So we're interested to see, can we use RNA sequencing to gain insight into this kind of economic exchange that's happening between the tree and the associated fungal species, and what happens when you disrupt or make a change to the nitrogen balance of the soil. So our experimental site is a forest called Flackaliden, which is about 40 kilometers inland of Umeå. And this is a site where for the past 25 years, during the growing season, every year, the site has been supplied with a balanced nutrient mix. And our particular interest is that this includes supplying nitrogen to the trees. And this is a nice experiment because it's one case where you absolutely don't have to struggle to see the effects of the treatment. If you walk into the control plots within this forest where no nutrient enrichment has occurred, this looks like a typical spruce forest in the north of Sweden. You have lots of bilberries on the ground, lots of light coming through the canopy. Whereas if you walk into the nutrient-enriched sites, it looks totally different. There's very little understory. You see ferns, but no bilberries, and the trees are very much bigger with a very dense closed canopy. And this has led to a significant increase in tree height, in above ground biomass, and also below ground biomass of the trees. So what we did is a long time ago now, in 2011, we sampled a number of weeks during the growing season, and the region here marked is showing when the nutrient enrichment treatment is applied during this year. And we went into the forest, dug up soil and isolated fine roots of Norway spruce trees. And then our sample that we're extracting RNA from is those roots and all of the associated fungal species around the root. So we extract RNA from that and perform standard Illumina RNA sequencing on this. And then we developed a pipeline where we first align reads to the Norway spruce genome that we have, and we can look at gene expression in the tree side. We take all of the reads that do not align, we assemble those, and we match them to available fungal genomes so we can look at gene expression in those fungal species. So an important caveat or thing to have in mind in this is that we're aligning those reads to the closest matched reference genome available. So we call it that species, but it's not necessarily always the case that the read comes from the exact species that is the closest match available. So for this project, all of the sequencing results that I'm presenting were generated at SciLifeLab. And for us, having access to NGI is really important because we get expert help and input from them on how to design and run the project. And they are infinitely patient with us in sending them sometimes not the best DNA or RNA and giving us feedback on how to improve this. And SciLife has always been brilliant at having access to the latest sequencing technologies. And for this particular project, really instrumental to the success of the project was that we had an NBIS WABI long-term bioinformatics support project. And that was really instrumental to developing the pipeline that we have used for this. So one of the first things that we did with our data was to count how many reads do we assign to each of the fungal species that we can identify using the RNA-seq data. And this allows us to look at who is there associated with the tree roots and to look at the difference in abundance of those species in our nutrient limited and nutrient enriched condition. So NL is nutrient limited and NE is our nutrient enriched. So this is our control condition. And looking in this PCA plot, we can see one of the biggest effects that we see is actually a change in abundance for some of these species. And I will highlight a few species throughout the presentation. So the overriding picture to come out of this was that we had a decreased abundance of Piloderma and Quaternaria species in response to nutrient enrichment, and an increased abundance in nutrient enrichment of particularly Cenococcum, which I will concentrate on. Another way of looking at this is to actually just plot the equivalent of a PCA. In this case, it's a method called FATE, where we're looking now at the individual transcripts from all of the species and the expression value of those transcripts. And this is a multi-dimensional scaling method. And you see this striking pattern that there are strands in this distribution. So we wanted to understand what's the basis of this. We color the genes by whether they have higher expression in nutrient limited or nutrient enriched conditions. And you can see that there's a separation based on whether they have higher or lower expression in the two conditions. And what we found was that you can look at the individual strands and pull out the transcripts within those. And there's a high enrichment within those strands for transcripts coming from different individual species. So each of these strands is largely representing an individual species response within the two conditions. And we can then pull out the transcripts specifically assigned to Piloderma, to Cortinarius, and to Xenococcum, and we can look at how do these look. And what we see is that in the case of Piloderma and Cortinarius, a majority of their genes have higher expression in the nutrient-limited condition throughout the growing season. And in the case of Xenococcum, it's far less responsive to the nutrient enrichment condition. So a majority of its genes basically do the same thing in the two conditions, but there is a specific pattern of enrichment for some genes in the nutrient enrichment condition. What we can also do is aggregate all of the data to the community level. And now we take all of the transcripts from all fungal species and we're plotting a PCA looking at the different time points that we sampled through the growing season. And in this PCA plot, we connect the control point for 1 week to the nutrient enriched point for the same week. And the picture that emerges for this, for the fungal species, is that there's no real seasonal pattern that we can identify from this PCA, but there's a very, very strong overriding signal of an effect of the nutrient enrichment condition. So the length of these lines between the control and the enriched point is very long, and this has a strong effect on the fungal community. And because this is expression data, we can do kind of standard, more systems biology style analysis. So we generated co-expression networks. And this particular figure is showing this at the community level. So we aggregate reads from all species, and we can identify sets of co-expressed genes within this co-expression network. And what we find is that there's a set of network modules that largely represent genes that have higher expression in nutrient-limited conditions and a set that have higher enrichment in nutrient-enriched conditions. We can look at their profile of expression through the season. And what we find is that if we look at these modules, there's enrichment for Piloderma and Cortinarius. And if we look at the ones with higher expression in the nutrient enrichment condition, we again see overrepresentation of transcripts from Xenococcum. We can also do exactly the same thing using or the same kind of analysis using our data from the tree side. So if we look at a PCA for all genes expressed in the tree, we see quite a different picture. So the length of these lines separating the control and the nutrient enriched condition is much shorter in the case of the tree. So the nutrient enrichment condition is having a smaller influence on gene expression on the tree side, but we see a very striking seasonal pattern with different expression profiles through the season, which is exactly what we would expect from tree roots, but there's been very little transcriptomics data generated from tree roots out in the field. And we've done the same kind of network co-expression analysis from this. And from this, we can then look to see through the season, what are the different kinds of biological processes that are enriched at the different time points and within these different network modules. And we can go in and look at these and find specific genes of interest as well. So for example, looking here at sugar efflux transporters. So these are sweet genes that are regulating the efflux of sugars from the root out into the soil. And we can see that there's higher expression of these, for example, in the nutrient-limited condition. And this is interesting because immediately we can hypothesize that in nutrient-limited conditions, there's higher expression of genes supplying carbon to the fungal species. So what we then have is our co-expression network on the tree side, and we generated co-expression networks for transcripts assigned to our 3 key species that we're interested in. And we wanted to use this to look at coordination between the 2 components of this system. So can we identify aspects of the co-expression network for one of our fungal species and how they're linked to changes in gene expression on the tree side? And what we find is that for Piloderma and Cortinarius, there's a higher number of network modules in their co-expression network that have correlated profiles through the season to network modules on the tree side in nutrient-limited conditions. Whereas for Senococcum, there's a larger number of network modules in Senococcum that are correlated to modules on the tree side in the nutrient-enriched conditions. So we can see that for these species, there's a shift in which species are coordinated in their metabolism, for example, on the fungal side with what's happening on the tree side. And then we can go and look at this in more detail and see what are the biological processes enriched in those modules. And more specifically, what we did was to predict fungal effector proteins. So these are small secreted proteins from the fungal side that are used to control and direct the interaction with the tree. And we can see, for example, on the fungal effectors in the nutrient-limited condition in Cortinarius and Piloderma, we have very much higher expression of these in the nutrient-limited condition and strong downregulation of these in the nutrient-enriched condition. And that contrasts to Senococcum, where the picture is more even between the two conditions, but with a slight increase in expression in nutrient-enriched conditions. And we can then take each of those fungal effector proteins and identify what are the genes on the tree side that have matched expression profiles through the growing season. So here we show the number of genes with correlated expression on the tree side, where the paler color in each case is representing the positive correlation, the darker is negative correlation, and we have nutrient enriched in the kind of brown color and nutrient limited in the green color. And we can go into sets of these genes and see what do we have here. So for example, we can see that there's positive correlation in the nutrient limited conditions between these fungal effectors from Cortinarius and again, the sweet proteins controlling sugar export from the tree side. So we can really dig down into individual specific molecular hypotheses that allow us to understand what is driving the interaction between fungal species and the tree, and what are the differences in that molecular communication between the fungi and tree that are induced by our nitrogen treatment. So really we can move from the molecular level up to understanding the ecosystem style response that we see in the system. So we've been using this data to address biodiversity questions and community composition questions of who is there. We are able to use the expression data to ask what are they doing for those who are present. We can look at how are the associated fungal species linked and coordinated to the tree and how does this relationship change. And we can go into more detail, for example, looking at specific molecular examples like these effector proteins to see what's driving that change in interaction at the molecular level. And so we've done this in nutrient-enriched conditions. We think this is a brilliant tool to be able to go out into the environment, sample this kind of information and understand how our fungal species or other microorganisms coordinated with tree-level responses. So I'd like to thank the key people involved in this. This was a project that's an interaction or a collaboration with Vaughan Hurry and Torgny Nesholm in SLU. And the majority of the work presented today was done by Alonso Serrano and a postdoc in Vaughan's group, Simon Law. The metatranscriptomics pipeline was initially developed by my PhD student, Andreas Snyder, and that was done in this long-term support project with NBIS with Johnson, who was really instrumental to this. So thanks for the opportunity to present this.
Thank you very much, Nathaniel, for that excellent presentation. We have one question in the chat, so if you like, you can address that, or we can come back to that also at the Q&A session at 11:45. Otherwise, I think this is a good time to take a short break, stretch your legs a little bit, and then we meet back at 11:15 for a presentation by Siv Andersson on the cellular complexity of bacteria. Welcome back, everybody. I hope you had chance to stretch your legs a little bit in the break. I would like to thank all the speakers for the first half of the session for very interesting presentations. And please gear up with questions for the Q&A session at just before lunch. We have two more very interesting research presentations before we go to the Q&A session. And first out is Siv Andersson. She is a professor at the Department of Cell and Molecular Biology at Uppsala University, among all other of her tasks as well. We very much look forward to her presentation on the evolution of cellular complexity. So please, Siv, go ahead.
Thank you. And let me share my presentation. So you can see it now? Good. So normally we work with bacterial symbionts, but today I'm going to speak about a very specific project that we have worked on for the past few years. And many people in Uppsala have probably seen the bacterium that I will talk about. And this is because this bacterium was selected for a public art project in Uppsala. So it was featured as a light show at the north entrance of Rosendal, which is a newly built area just near to SciLifeLab in Uppsala. So here you can see this beautiful bacterium. Some of you may even have walked on it, like this little child to the right. And today you will hear a bit about the science behind the bacterium. So this bacterium is interesting because if you have a quick glance at it, it's not immediately clear if it belongs to the eukaryotes and the prokaryotes. And you're all familiar with this picture. You know that eukaryotes have internal membranes, mitochondria, nuclear membrane, and so on. Whereas prokaryotes, which consist of bacteria and archaea, do not have such a structured internal cytoplasm. Instead, bacteria have a very thick peptidoglycan cell wall. So these bacteria that I will talk about today belong to a group called the Planktomycetes, the mysterious cells. And if you look to the left, then you can see that they do have internal membranes, a bit like eukaryotic cells. And the type species is Schemata obscuriglobus, and it's so called because it is an obscure globular cell. And these cells are also a bit atypical in that they can do things that normally bacteria cannot do, such as, for example, importing big macromolecules such as proteins. And to the right here, there is a recently described species, Microbial Single, which can even import and take up entire bacterial cells in a process that seems similar to phagocytosis in eukaryotes. And before I saw this, I have never heard of a bacterium that is able to take up another bacterium. So what are these cells? When the people first looked at them in the microscope a long time ago, and I should say also that they are present in soil and in waters, they thought that they were simple forms of fungi. So that's why they have this strange name, Mycetes, in their sort of the Planktomycetes, the planktonic small fungi. Then they realized they were not fungi, and for a while they were thought to be some kind of intermediate of prokaryotes and eukaryotes, and some people even speculated that they were the ancestors of the eukaryotic cells. More recently, people now think that they are bacteria, and it has been suggested that they may have gained some genes from the eukaryotes. So it was interesting really to look at their genomes to see what do they look like, and can we identify any genes that they have gained from the eukaryotes. So several groups tried to sequence this genome and failed to. They could produce sequence data, but it was very difficult to close the genomes. And then we decided also to give it a try. And when I say we, I mean in addition to myself, my PhD student, Mayank Mahadyan, who did the Bioinformatics, and my postdoc, Benjamin Yi, who did his PhD studies in Australia with John First, who was one of the pioneers who studied Schemata obscuriglobus. So we decided to try to sequence their genomes. And at first we also failed. And then the PacBio long-read sequencing technology came along, and voila, now the genome assemblies that Mayank did, they worked just perfectly, and we could assemble these genomes and take a look at them. And the genomes are, for Schemata obscuriglobus, 9.2 megabase pairs. We also sequenced a few relatives, and they also have genomes in the 7 to 10 megabase pair range, which is at the upper range of bacterial genomes. When we analyzed them, we found that they were clearly bacterial genomes, but we could not detect any massive gain of genes from the eukaryotes. Rather, it seemed that they had expanded their gene repertoire by duplication and domain shuffling, and we found many new multi-domain protein architectures. In several cases, we saw proteins that had connected domains that are common in prokaryotes with domains that are common in eukaryotes. And there was expansion in post-translation modification, signal transduction, secretion, and transcription regulation, functions that you would expect to expand in a cell that has a complex internal structure. Still, this did not really tell us why did these cells, these bacteria, have internal membranes, and how did these membranes originate. So we decided to also take a closer look at the bacterium to see what they look like. And for these studies, then Christian Seeger, a researcher in the group, stepped in and has been responsible for the microscopy studies together with the scientists at SciLifeLab. So when we started, then the view of the cell plan was that these bacterial cells, they contain an outer membrane, they contain a cell wall, they have these intracellular membranes, and it was also suggested that Gemata obscuriglobuus has a nuclear-like membrane. But this was also controversial, and there has been a big debate in the field as to what exactly do these cells look like. So we took a look, and it was very clear from all the images that we have produced that they do have these internal membranes, but the structures, they look a bit different from species to species. Still, from just 2 dimensions, it's very difficult to understand exactly how these membranes are connected to each other. So we also wanted to do a 3-dimensional reconstructions. And this was done with the aid of FIB-SEM tomography at the Umeå core facility for electron microscopy. And what they did was quite remarkable, I thought. They took this bacterial cell and then they sliced it into about 100 different segments. And then they took scanning electron microscopy micrographs of each individual slide. And by putting all these images together, we could then reconstruct the 3-dimension of these bacterial cells. So we did this for Tuvongella immobilis, and we also did it for Gemata obscuriglobuus. And Gemata obscuriglobuus has a condensed DNA. So in this case, we could even see the DNA, and you can see it here as this green part inside the cell. And in this B section, we have a cell that is actually in the process of budding. And I will now show you a video of how all these 100 slices and micrographs can be put together, so you can get a feeling for how the cell looks like in 3 dimensions. And once you see these images and how after that we put it together into a 3-dimensional cell model of what the cell looks like. And by looking at this 3-dimensional cell model, and we reconstructed it for a few different cells of both Tuvongella and Gemata obscuriglobuus, we saw that there were clearly membrane invaginations, but there was no nuclear-like membrane that really enclosed the DNA. Rather, it looked like the cell is a cave with many connected tunnels. So it has this very complex internal membrane structure, but the cytoplasmic space is sort of connected all through the cell. So then after that, we concluded that it does not look as if they have a nuclear membrane, so we ruled out that hypothesis. So what about the cell wall? When the old school microbiologists looked at these cells in the mid-1980s, they thought that the cell wall consisted of proteins. But more recent studies, 4 or 5 years ago, it was concluded that the cell wall is a typical bacterial peptidoglycan cell wall. So we said we had to also look at the cell envelope and see what that looks like. And to the left here, you see a model of a typical cell envelope like in E. coli, where you have an outer membrane, you have an inner membrane, and then in between you have this peptidoglycan cell wall. And then there are a number of outer surface proteins and transenvelope spanning complexes. And the distance between the inner and outer membrane in E. coli is 25 nanometers. And this is very important that this exact distance for all processes to function properly. And then in Gemata, using now cryo-electron tomography at the SciLifeLab Cryo-EM node in Umeå, we got these beautiful images, and you can really here see both the outer membrane and also the inner membrane. And the inner membrane is not always in close contact to the outer membrane, but at places where it is, the distance is sort of variable, but on the average about 45 to 47 nanometers. And at first we wondered, would it be possible even for complexes to cross, go across this outer and inner membrane? And luckily the resolution of these images were so good that we could even look at individual complexes. So we could free, for example, here to the left, you can see these hats marked in yellow, and that are outer surface proteins. And then in the middle, we have pili that marked in red that really stretches across from the outer to the inner membrane and connects them. And if you look at this image of the whole cell, you see these little yellow and red dots, which are all these structures, and they are all over throughout the sort of cell envelope. Still, this did not really tell us about sort of the composition of the cell wall. So we decided to also isolate the cell wall to see exactly what does the cell wall look like. So here is an electron microscopy study of the cell wall done at Huddinge, the microscopy unit in Huddinge. And you see these remarkable features here in the middle. So it looks like a balloon that is filled with these holes, pores, and we hypothesized that the pores may correspond to the site where we have these trans-envelope spanning complexes or outer surface proteins. So then we took this balloon-like cell wall and sent it to the proteomics facility. And they analyzed the proteome of the cell wall, and they came back to us. And we found that the cell wall mostly consisted of two completely novel types of proteins with YTV motifs. And the YTV motif, which is marked here in these little green dots, it is the only PIF domain that is unique and also conserved among all taxa in this Plankton mycetalis. And they are cysteine-rich proteins, and this means that the stability of the cell wall can be modulated by making and breaking disulfide bonds. We still don't know exactly how these proteins form the cell wall, but it's quite clear that this is really a unique feature of the Planktomycetes that we have not seen in any other bacteria. So then what about the peptidoglycan cell wall that other people thought it contained? We also looked into the genome, and to make a peptidoglycan and also to synthesize it and then elongate the whole bacterial cell and finally divide it, it requires a lot of genes. And when we looked into the genome, it was clear that all these genes had disappeared and been lost. So they do not even have the genes to make a peptidoglycan cell wall. So from that, our current sort of model is that these bacteria, they have an outer membrane. They have intercellular membranes that are invaginations of the inner cytoplasmic membrane. They have no nuclear membrane, they have no peptidoglycan cell wall, but instead they have a unique protein-rich cell wall. And we believe that this remodeling of the bacterial cell wall was a key event in the evolutionary history of these bacteria. And it had a number of morphological consequences. First of all, that the cells become spherical and they start dividing by budding. And it also enabled membrane invaginations. When you have a rigid peptidoglycan cell wall, it's not really possible to have these membrane invaginations. And the beneficial effects that we can think of is that this enables the cell to import large macromolecules, such as big proteins, also large carbohydrates. And they can then have a larger periplasmic space for storing and degrading these macromolecules. So we think that selection has been for large predatory selfish cells, and this can be beneficial when you are in an environment when the nutrients come in sporadically. So suddenly there's a lot of nutrients, and of course if you're a big cell and you ingest all these nutrients inside the cell, and then you can slowly degrade them during periods of starvation. So we think that's why these cells have originated from normal bacterial cells, and then they have got these internal membranes. Some people speculate that they are the ancestors of eukaryotic cells. We do not think so, but we think that by studying these bacteria, we may learn something about the processes whereby cellular complexity originates, and maybe the process also whereby cellular complexity originated in the eukaryotes. And finally, thank you all the people in my group. I've already mentioned Mayank, Benjamin, and Christian, and also all the facilities that we have utilized. We have, for us, it has really been like a smorgasbord where we have cherry-picked whatever we have needed for the moment, the Uppsala Genome Center, the proteomics facility, and the centers for electron microscopy we have utilized both in Umeå and in Huddinge. And this work was supported by the Knut Nalles Wallenberg Foundation and the Swedish Research Council. And thank you for listening.
Thank you very much, Steve, for that incredibly interesting presentation. Very interesting scientific question, of course, also a very good example of how different types of services provided by SciLifeLab can be combined. Perhaps we can take one question before we move on to the next speaker. You can post a question in the chat or just unmute yourself and ask a question if you'd like to do so. Maybe I can have just, oh, was there a question? No, then I can just ask. Yeah, yes, sure. Go ahead, Christoph.
Maybe I can try to ask a question. Can you repeat how big were the genomes of Plantomyces and were they substantially bigger than what is usually found in prokaryota, in bacteria?
So the genomes are, they are in the upper range, 7 to 10 megabase pairs. And there are also other bacteria that can have such large genomes, but it's not kind of atypically large. It's still within the bacterial range, but certainly in the upper range. And for several of the domains, they are really at the top. They have some more domains than many other bacteria, or particular specific domains.
Okay, thank you.
All right. Thank you very much for that question, Christoph. So I guess then we can just slide over to the Q&A session, which is the last part of this session, where we can revisit all the presentations and all the speakers are welcome. We will also welcome Sebastian Di Lorenzo, who is a community coordinator at the SciLifeLab Bioinformatics platform, the National Bioinformatics Infrastructure Sweden, that can address specific question relating to the bioinformatics services.
Hello.
So while everybody is joining, I think there's one question we can address, and we can send it direct to Sebastian, and that's the question about long-term support. So as you see, many researchers here and also in the community have used long-term support. So perhaps you can expand a little bit on that concept.
Yeah, it's a very successful Wallenberg-funded initiative that we've had for many years now. So NBIS is divided into this long-term support and short-term support tracks, basically, where the short-term support you can get help with. You can just apply for it, and then we find a bioinformatician, and you pay as you go. You're paid per hour, but the long-term support is fully funded. So there you apply, and we look at the scientific feasibility and what kind of samples you have, and we look much more closely at it and compare it with other projects that searched for support in that round. And then we just assign a bioinformatician for a much longer period of time, about 500 hours. So it's a very close collaboration with the group, and you're really embedded in that group for a longer period, and fantastic work results from it.
So I think it's a good service to take note of. Can be very valuable. We actually have one question from earlier in the session for Nathaniel that I think we can address. It's from Stefan Pettersson, and he asks about ectomycorrhizal interactions, if that's less significant under high nutrient regimes. That's sort of a prevailing view. So he asks if there's a way to have more quantitative insights about this from the type of sequence data that you have generated, Nathaniel. And also, do you have internal?
Maybe I can just add a little bit. So,
sure.
I mean, it's basically coming from the tradition of working with plankton, right? And how people are starting to more routinely use internal exogenous standards that you add to any kind of transcriptome experiments to not only say some things about relative expression levels, but actually absolute expression levels per volume of water or per gram of soil or whatever else. And everything, what are they you can think of. So I'm just thinking if you have been considering to do these things as well, because particularly here with these kind of mycorrhizal interactions, there is this idea that this gets less important when you have a more nutrient kind of enriched environment, right? So potentially you could use your data to also say something about that, and not only the relative quantification of gene expression. You can actually say if something is upregulated or just relative some other functions.
Yeah, I can say we've absolutely thought about the need for this, but we haven't come up with a technical solution that we think we can apply for this, because we absolutely have this confounding issue of abundance of the species with the apparent expression level that we see. And at the moment, we're using ITS data to kind of compare this to, so amplicon data, so we can see, do we have independent confirmation of whether there is a change in abundance of the species as well. But no, right now we haven't found a good technical solution to do that. But it's a huge limitation in the analysis we're doing, that we can only ever discuss in relative terms.
I think that from the biological oceanography, there might be some inspiration, but of course you have the added complexity when you deal with sort of a soil and a structured environment there. Yeah. So I see the need for perhaps some method development in that field. So
absolutely, yes, I agree.
Okay. We also have a question for Siv here on planktomycetes. So it's a question about the potential of planktomyces as a novel source of bioactive molecules.
Yes, and I'm glad you're asking this question, because I forgot to say that the planktomycetes are currently, many people are interested in them because of this, that they produce bioactive molecules. So there's a lot of searches for them in the environment, because obviously they probably send out, export bioactive molecules to kill all their surrounding cells, and then they eat up all their proteins and carbohydrates and everything. So many people go out and search for them for this specific purpose, to find new bioactive molecules. So I think it's potentially a very rich source of new antimicrobials.
Okay, thank you very much, Siv. So, the floor is open. Anyone can ask questions either through the chat, or you can just unmute yourself. You can ask questions, of course, relating to the specific topics that was raised in the presentations, and also about their interaction with SciLifeLab. How did they do that, and what services did they use, and what did they think about that value? So the floor is open. Anyone who has any specific questions?
Hello.
Yes, you're welcome.
Yes. So I just wanted to repeat the question, which was shortly touched by Olga, about medical questions compared to these more biological questions. Is there a way how SciLife prioritizes their resources? Because it came up in the introduction, so I was interested in that.
Yes, thank you very much for that question. So the majority of infrastructures that SciLife has is actually able to cover any aspect of biology, if you perhaps exclude facilities like drug discovery and the clinical sequencing. Other than that, SciLifeLab does not have any prioritization of the projects, and we serve everybody on first come, first serve basis. And we do not divide different queues. So the only thing that we're prioritizing right now is COVID sequencing.
Okay.
That is purely due to the current conditions, but otherwise, no, we treat all the projects alike. And actually, even to enforce the necessity of engagement of SciLifeLab infrastructure with the wide scientific community, so not just medical, but everybody else, then there is the new capability of planetary biology, actually, that is supposed to work on the strategies. How can we engage the biodiversity and evolutionary researchers in conducting the world-leading research using the SciLifeLab infrastructure? So it's exciting development. We'll see how it goes.
Okay, well, no, that is good to know. I think it's just when I myself discuss with others, you do hear this bias, or like this idea of people that they say, well, we shouldn't. I work myself with biogas research, and then but that's why I wanted to double check how that is.
Everybody is welcome. So you're most welcome to contact us, and we'll take care of you. Either you're working with cancer or you're working with parasitic worms. So at the facilities, we basically do not care what you do. We just want you to do a good job with the data that we produce.
Okay, good. Thank you.
Most welcome.
Any comments from any of the speakers on this issue on getting access to SciLifeLab services and interacting with SciLifeLab?
Can I comment?
Sure.
Yeah, so I think it's so everything Olga has been saying in the presentation is true. So if you're about to embark on some new project that involves sequencing, and you're not sure, you haven't done exactly that before, it's a really good idea to contact them and like pick their brains and get up-to-date advice. This is exactly what we did. So, and we have been doing that often for different plant species even, because I mean, sometimes it's easier than other times to get really good quality DNA. And then you want to discuss with somebody before you decide, okay, these are the constraints. How can we do the best possible job with these constraints? And I would strongly recommend to contact the platforms ahead of time. Also, even when you're writing a grant proposal, you can contact. I was in contact with the annotation platform before I wrote my ERC proposal, and that really helped. So yeah, they're here to help.
Thank you, Tanya, for that comment. Anyone else who would like to chip in on that discussion? I think it's fairly central to this session. No?
I can only say that I completely agree with everything. That's exactly our experience as well. I mean, I'm infinitely thankful to Olga. She's always helping us with advice on DNA extraction and things. And it's brilliant to be able to contact before you get going to know, is this going to be feasible or not? And I think that's really what differentiates SciLife from commercial services that are available.
I think that's also it's important to point out that it is the custom capacity of SciLifeLab is really what stands out and what we value, that we are willing to sort of engage and develop our methods together with users. Perhaps one or two more last questions from the audience before we wrap up the session. Any more things on your mind? Otherwise, you are also, of course, very welcome to reach out to us. You can reach SciLifeLab, and the scientists have been presenting here through many channels. You can just start anywhere, and then we will guide you to the right type of facility or service that you might require. So with that, I would just to wrap up and give a big thanks to all the speakers and for taking the time to present their work and their opinion on working with SciLifeLab, and for all of you in the audience who have listened in and asked questions, and hopefully gained some sort of ideas and thoughts about how to further your own science. So thank you very much, and have a nice lunch.